I want to thank Ronse for giving me the opportunity to speak here. It's really a privilege, and Anil for introducing this session. And it's really fun to speak after Liz because we have collaborated, and I actually included a slide from a paper we published together. So it's really fun to be here. All right, so okay, I have to share my slides. All right, we are good. So the numbers, a lot of you out here know this, and that's why we do this. But I do want to start with the numbers. So about 230,000 women get diagnosed with ovarian cancer worldwide, and approximately 140,000 don't make it. It's a really high number. At least when I first saw this, it was astounding to me. And the other point I want to make is that ovarian cancer is the most lethal gynecological malignancy, particularly in the Western world. And this is something I don't think is appreciated enough. We hear a lot about different cancers and how bad they are, but the fact that ovarian cancer is the most lethal gynecological malignancy is something which really needs to be noted. So the number that got me into this particular project, which I'm going to talk to you about, is on this slide. So it starts with the fact that most women come to the clinic, and folks like Liz and Anil treat them with disease that has spread, which is basically metastasized distant disease, and the survival rate is below 30%. But if serendipitously they came to the clinic, and this obviously happens, when the disease has not spread, it's localized, and we classify that as stage 1 or stage 2 disease. Basically, it's a 92% survival rate. To me, this is mind-boggling that we can go from 28.8 to 92 by just detecting this disease early. It's not incurable, and I don't think any drug is ever going to move the dial that much. So early detection to me is really something which is super critical for the field. So what do we have? So unfortunately, this is an area where the progress has been limited. It's not easy, and it's just been stagnant for a while. CA125, which again, a lot of you know about, is the only sort of blood biomarker, and there's a few others, but it doesn't work for early detection. It has other applications, and therefore there is no early detection screening program for ovarian cancer. You cannot get anything. Imaging does not work. There is no mammogram. And this is the scary part. Right now, except symptoms, there is no way to understand or know that somebody has ovarian cancer. And I think this really needs to be understood very clearly by the field. And we have to look beyond traditional biomarkers. What I mean by that is, you'll see in a minute, there's a lot of new discovery, new molecules that are coming into the field of biology, molecular biology, and that has to be harnessed by folks working on ovarian cancer. And that's what I'm trying to attempt to do, is that we really need to think outside the box. We can't keep doing the same things we did for the last few decades. So this is a slide I showed to the first year grad students when I used to teach. Thank goodness I don't have to do that very much now. And this slide basically tells you that 2% of our genome makes proteins, protein coding genes. What does that mean? So every gene that you hear of in almost in the mainstream, where you hear of drug targets, you hear of the BRCA genes that Liz just talked about, every gene that you think of, and not just in cancer, in other human disease, diabetes, they come from this 2% of our genome. So 60 plus years of molecular biology focused on 2% of the genome. And for that, for decades, and like when I went to grad school, and I will admit that was a long time ago, we were told that the rest of the genome, 98% of the genome, was junk DNA. We need to step back, as a species, as a civilization, to think that we think nature is so frivolous that it would make 98% of junk DNA, and it's only 2% of it is going to be useful. So it's completely wrong, as you can imagine. And now we know that the rest of the genome, 80% of the genome is making what we call non-coding RNA, RNA that is not making protein. Thanks to COVID, that's a weird phrase, but thanks to COVID, everybody knows about RNA now. And I get asked this all the time. Hey, you work on RNA. COVID is an RNA virus, and then there's a pause where somebody expects me to connect the dots. I mean, I say there is no connection. That's it. I work on RNA, and COVID is an RNA virus. But this species, these non-coding RNAs, it's the tip of the iceberg. We really don't understand a lot about how they impact anything, how they impact our cells, how they impact cancer. So this is a tremendously exciting area of research. Today, I'm going to talk about one class of non-coding RNAs, one class of molecules called microRNAs that affect the protein production. And this, how we can harness this, how we can use this for early detection. So a little bit of background, you don't have to go into the details, but I just want to get you a sense of numbers. So microRNAs exactly as the name sounds, are these tiny bits relative to the mRNAs which make protein. These are tiny little bits of RNA which are everywhere, as you will see in a minute. But just to look at numbers, there are about 360,000 mRNA molecules made in our body, in our cells. These are the mRNA which go on to make protein. So the COVID-19 vaccine that you're getting, hopefully, well, this is Texas, I don't know, but hopefully in the rest of the country, that is making, that's an mRNA that's going to make the protein, giving you the antibody reaction. MicroRNAs, there are only 2,500 different microRNAs. So the numbers are low. And what we like to believe is that a small number can actually summarize a lot of information, genetic information that can be conveyed. And that's really what I want to emphasize. And just an importance, so this slide is already about 10 years old, but I still like it because every stage of cancer that we have looked at, from the genesis, like the start, onset of cancer, which is basically when the cells are proliferating, dividing in an unregulated manner, to tissue invasion, metastases, when it's spread, to sustained angiogenesis, which Liz mentioned, basically that the cancer can grow and get nutrients, and cancer therapy. Every stage, microRNAs have been shown to be involved in a very visceral, very direct way. So that's how important this completely new class of molecules. So literally 20 years ago, nobody in the cancer world, nobody in the mammalian world knew about this. And now we know it's involved in almost every human disease and all the steps that we can study. So how is the connection? This is all background. How is this connected to the work we do in the lab? So my lab actually works on the BRCA genes. We work on genome stability. That's the basic science. We study why is BRCA genes and how do they function, and PARP inhibitors, which Liz already described. But we found the first microRNA that targets the BRCA gene. So what does that mean? So as Liz said, we sequence the genome to understand the defects in the BRCA gene. What we showed was that even if your BRCA gene is intact, if you actually make more of a microRNA that suppresses a BRCA, the phenotype, the ultimate result will be like a BRCA loss. And what was really pleasant, or I would say rewarding for me, is that after our paper, there was a paper published showing that the microRNA we found, MIR182, actually does that in ovarian patients, where more 182 correlates with less BRCA production, which is equivalent to basically saying that you have a defect in the BRCA pathway. So this is another way that a tumor can use these machineries to basically create problems, allowing it to grow. So this is a completely new area in that way. So we have continued over the years to work on microRNAs. Again, I don't want to take too much time on this, but what we showed here was one microRNA, 622, specifically in BRCA1 mutant cells. And see, this is a PARP inhibitor. So this one microRNA impacts the effect of the PARP inhibitor on BRCA tumors. So that's one. The second story, which I'm excited to present, was actually with Liz. This is the slide I included, that with tumors that we got from the Swisher lab, we sequenced them, which means we looked at every microRNA in them and tried to find microRNAs that impact the patient's survival and therapy. And again, without going into details, I want to just point out this one graph to you. So this is one microRNA, 4935P, and this is BRCA2 mutant tumors. Look at the astounding difference in survival. So if the patient has low levels of this one microRNA, the survival is here. If the patient is making high levels of this, it's really profoundly impacting survival. You know, we don't completely understand how, and to me, it's still mind-boggling that a tiny piece of RNA has such a profound impact. or potentially direct impact in patient survival. So that is not even what I'm going to talk about. I'm going to talk about circulating microRNAs. How can they be used as biomarkers, non-invasive biomarkers, blood tests? So it turns out, again, new field, but in the last 10 years, there's a lot of work showing that microRNAs are in our blood. So about 10% of the total microRNAs. So as I said, there are about 2,500. So imagine 200 to 250 microRNAs are always in our blood, like your cholesterol, like your glucose, they are in circulation. And the way we want to think about is that a small subset of them, any changes can teach us about the pathology because they are in circulation. Can we use the microRNAs like a GPS where we don't have to go deep before, like we cannot with every patient, but just to, as a blood test, as a biomarker, can we figure out something is wrong by using this new class of molecules? And they are not, from just purely logistics, they are easy to use because you can isolate them from blood relatively easily. You don't need a lot. Quick analysis time, and something which is super important, they're stable. So, we get samples from India, we get samples from Poland. It doesn't matter. You do not need a fancy lab in the US to make this for it to work. It can work wherever the blood is drawn. It is that easy. And then it's very sensitive, which means you can amplify it. So you don't need to worry about it getting lost. So those are logistics, but super important if you really want to use it. So the two people, this is the most important slide. So the people who really did the work, I'm just talking about it, is Kevin Elias, is a surgeon here at Brigham. He's an assistant professor here, has his own lab. Kevin has really started this project with me because we were doing the microRNAs for other things, and he really started it with me. And Wojciech, he was an ex-trainee, really brilliant guy who's back in Poland, has a big group who does the statistics, and this is all about statistics. So these two guys are the key to this project. So what we asked is, could microRNAs help us diagnose cancer, ovarian cancer? And we actually showed that a few years back. And I want to show you unpublished data, so I want to quickly go over this. But what this paper showed is that with a blood test, we can detect ovarian cancer with some accuracy. See, the big question is, can we detect it early enough? So that's what I want to go towards. But this was very important as a proof of concept that we can do a blood test fairly easily to detect with basically over 90% accuracy that somebody has ovarian cancer or not. So the way the test works is basically we take a vial of blood, like a lot of people go for a physical, you isolate the RNA, again, nothing too complicated. For the development of the test, we sequence. For actual application as a diagnostic, it would probably be PCR, but it's not too complicated. But at this stage, we had to find every microRNA. That's why we did it. And then the analysis. This is the novelty. So this is the cool machine learning, artificial intelligence. This is exactly how Netflix knows what shows you want to watch. Amazon sends you orders even if you have ordered them. So that concept. But it really, truly, it's statistics. And I'll try to get to it in a minute. So it's what we call the neural network analysis. And the analogy I like to give is it's, when we sequence blood, it looks like a night sky. This is just a whole bunch of dots and you have no idea what's going on. But we are trying to sequence blood from cancer versus no cancer. And then what starts appearing is a constellation, so a pattern. So once you know what to look for, you can find that pattern from anywhere in the world. That's basically the goal here. The machine learning approach gives us this pattern. And then how accurate this pattern is, we keep doing it over and over again, and the pattern gets solidified. Again, it's really global, so you can go and look at it from anywhere in the world. So that's the crux of this discovery. So in this first story, we could show that, with a really limited number of samples, that we could detect ovarian cancer with about 92% sensitivity, specificity. And just as a straw man, and I'm sorry, I know people know that CA-125 doesn't work, but that's all we had as a comparison. It's really much better or more efficient than CA-125. So that was basically what we started with. And one very important point that we could show, this is with public data, that this test, this particular subset of microRNAs, this particular pattern is specific to ovarian cancer. So we looked at 12 other malignancies and we could say that this is really specific to ovarian cancer. And what I would like to add is what is exciting is we can do the same for other cancers. We have a pattern for pancreatic cancer. We are developing a pattern for breast cancer. So this technology theoretically can be applied to other malignancies. But what we have and what we have really gone ahead with was a pattern for ovarian cancer. So one thing we also know that machine learning often doesn't work. Often you get recommendations for shows which make no sense to me, at least I get. And I wonder whether I made a mistake or the machine did. Well, most times probably I did. But to really know that it works, you need experimental biology. So here is what it is. What we could do then is go back to a patient who had surgery and look at the pattern. So literally 3 days after surgery, if we do the same blood test, we see that the signal is gone or going away, at least for a bulk of those microRNAs. So that is very important. That really tells us that what we saw, the signal came from the tumor. It wasn't random, it wasn't background. This is very important with any kind of data science approach where you need, I believe you need more experimental validation. And then the key question, that is the key to this, is how early? How early is early? Because the key is to detect it early. What we did was we could detect it. Now, detecting is important, but it is still may not be good enough. The patient has full-blown cancer. What will you do? So what we really need to do is figure out how early can we detect it. And one of the clues which was really pleasing for us is we could detect these microRNAs in early stage ovarian cancer. For the aficionados, it's the stick lesions, which is really the stage where it's considered the earliest stage of high-grade serous ovarian cancer. And in those tumors, this is actual patient tumors where we could stain, we could look at the microRNAs physically and see that they're present in early stage tumors, giving us some hope that this is something which is occurring very early and we can try to detect it early. So after this story, and I will open those black boxes in a minute, what we did was we collected a lot more samples, particularly focusing on early stage as I did, and we projected what is the capacity of our test. And if you project it, it's sobering. On one hand, this looks great. So we can have a specificity, accuracy almost of 99.1, which sounds good on paper. But in reality, for the general population, it's not good enough. Here is why. So if you think of 100,000 women, we will miss 22 cancers. So that is not a big deal. I mean, right now there is nothing to detect it. So 22 cancers is not a big deal. But what is a big deal is the false discovery. So 890 women will be told they have cancer when they don't. And that's been the real bane of this field with CA-125 and imaging. That we cannot, because the only option then would be surgery. So this number is way too high, and this number has to go up. But I really believe this is a matter of numbers. This is statistics. So if we get enough samples, if we can make our algorithm more robust, we can drive this number to 99.9. That's where it needs to be to even get close to the clinic. So that's where we are with this part of the project. But this also led us to a different side, which is the high risk population. So the BRCA carriers. They have a much higher probability of ovarian cancer, as we know. So can we focus this test on them and see whether we can get to the clinical application soon? So that's basically what we have done in the last few years, and I want to take you through this. So basically what we did is we collected as many BRCA samples. We actually have more. So we have sequenced over 400 BRCA carriers, and with disease, without disease from various sources, and we have a lot more coming. And what was striking, this is a super exciting result for me. Just look at healthy women. So if you are from a high-risk family versus not, your microRNA profile, the pattern of microRNAs in your blood are very different. So the defect in these genes is changing how the microRNAs look in blood. That's one. Second is if you are an ovarian cancer patient, with a BRCA defect versus no BRCA defect, again, the pattern is different. So having this risk really changes the microRNA profiles in your blood, something which can be easily tested. So Liz talked about the testing, and her lab has been a pioneer in the BRCA, like trying to come up with genetic testing. And what that does is you are sequencing a whole bunch of genes. And as she said, also sporadic when the genes are mutated only in the tumor. So they are sequencing the tumor. What we are doing is just looking at blood. So what is exciting is just looking at blood and 11 microRNAs, we are being able to predict BRCA status with about 100% sensitivity. And the accuracy is 94%, but I have a lot to say on this, which I will not here. What I will try to tell you is this could be a very important complementary test for knowing who's high risk, particularly in countries like India, where the BRCA testing is still prohibitively expensive and not feasible. This is a very cheap test as a surrogate to start. So this is probably the most exciting result for me, or one of them at least. I get excited about all results. But this is what we started this project for. Can we detect cancer in a BRCA carrier over a BRCA carrier who doesn't? So all these families are living in extreme anxiety that, can I, particularly younger women who have not had surgery, who have not had their ovaries or fallopian tubes removed, do I have cancer or can I have cancer? So this was the test. Basically, we took BRCA carriers with and without cancer and looked at their microRNA profiles. And we could, with 99.8% specificity, say you have cancer and you don't. This is obviously with old samples, archival samples. So if you project this like I did before, basically in 1,000 women, if 10 have cancer, that's sort of the proportion. We can catch 9 out of 10 cancers, which is great. And you miss one, but it's better than nothing. And 17 healthy women will be misdiagnosed. But the bar is a little lower here, because in this country, particularly in the West, almost all BRCA carriers or high-risk family members are told to have surgery. So all these women are going to have surgery. So 17 of them out of the 1,000 will be told to have surgery earlier, which is, again, it's not great, but anyway, the ultimate outcome was surgery. But this really got us closer to the clinic, where if we can detect it with that sort of accuracy, there is some real chance of application. The other piece of data which I'll share, which I think is really promising, it's very early and it's complicated, but I will try to go through it. What this is what we call lead time. So almost all the data generated for us was basically when a person came to the hospital with and without disease. So one right at the point of disease. But for few samples, from Poland, we got what is called longitudinal samples years before there was detectable disease. And that's what we are doing now. So what I can show you here is that we could see the signal versus control at least 2 years before detectable disease. That is very promising, because if we can detect the disease, if this pans out, again, it's very early, so I'm not going to, belabor this point, but the goal is if we can really detect the disease 2 years before it is detectable by any other method, there is real opportunity to cure. So that's why this is exciting and what we are doing. So this has all led to a clinical study. I'm very excited because this is the first clinical study based completely on the basic research in my lab, which is called the microRNA detection study at the Farber, where we are, and these are the, I would encourage people to go to our website and check out the protocol. But the bottom line is high-risk families who have been coming to our institute, we are getting blood from them, from a lot of these patients, women actually who are not thankfully patients yet, just come to get checked every 6 months. And the goal is to check our microRNAs in a way where we will really know whether, and if some of them develop cancer, unfortunately, we will really know how quickly we predicted it, because this is a 5-year study. So this is ongoing. And we are, because of one, again, mobile phlebotomy has become, blood drawing has become quite accessible now. So we are trying to get samples from all over the country, including the world. And this is, again, a very exciting progress for us. Now we have commitment from 9 groups in 5 countries where samples are, these are retrospective, again, old samples which are being given to us from BRCA carriers to validate what we have, because we've done it in 400 or 600, but this will let us, the numbers increase and really make our tests more robust. I've just put down the different sources and you will see it's really exciting. This is coming from Montreal, there is from England, which I'll talk in a minute, and all over the US. Folks who have talked to have been really generous with sending us blood serum samples to do our analysis. And I'll end with this slide because this is the most exciting set of data samples we have now access to. This was a huge study done in the UK where 5,000 women were classified as high risk based on their mutations or family history. 35,000 samples of blood have been collected over a long period of 7 to 10 years or something like that. And we now have access to this. So why this is so powerful is years before they had any detectable breast or ovarian cancer, the blood was collected. And we are getting that blood to test whether our microRNA is really predicted. And we are developing the microRNAs for breast cancer from this, which it would be wonderful to tell somebody with high risk that you don't have breast or ovarian and not limited. So I will end here, and I will acknowledge again the folks who did the work, Wojciech and his team. Dan Cramer, who's a leader in the epidemiology field of GYN, gave us early samples, and Kevin, I mentioned already. So I think we are supposed to take questions in the panel discussion, but thank you again for being here and for giving me the opportunity to present.